DNA consists of adenine, thymine, cytosine, and guanine. Each of them consists of a deoxyribose sugar as well as a nitrogenous base and a phosphate, and they all bind together using hydrogen bonds. And helicase unwinds them by breaking the hydrogen bonds, whereas topoisomerase ensures that the DNA remains stable. Replication is the process of replicating DNA to synthesize more DNA. So first off, the primase adds an RNA primer as the initiation complex, and following that, the DNA polymerase adds DNA nucleotides in the 5' to 3' direction, which is why there is a need for Okazaki fragments in the lagging strand, and in the leading strand, it's continuous. So in the leading strand, it is added next to the RNA primer. In eukaryotes, this occurs in the nucleus, whereas in prokaryotes, this occurs in the cytoplasm. So as the DNA is continuously added, the helicase continually unwinds the DNA, and topoisomerase ensures that it doesn't fragment due to stress. And in the lagging strand, there are segments called Okazaki fragments, which are synthesized separately by the DNA polymerase, and that is attached to multiple RNA primers, which need to be there because DNA polymerase can only synthesize from the 5' to 3' direction. So the Okazaki fragments are added next to the DNA primer, and following that, ligase removes the primer and replaces it with DNA nucleotides. And this is how DNA replication is done. And it's called semi-conserved because one of it acts as a template strand and the other strand is synthesized anew.
Transcription is the process by which genetic information from DNA is copied into mRNA, a single-stranded molecule that will later guide protein synthesis. This process occurs in the nucleus of eukaryotic cells and the cytoplasm of prokaryotic cells. In eukaryotes, a GTP cap is added to the 5' end of the mRNA during transcription to protect it and aid it in translation, while prokaryotes lack this modification. To begin transcription, the enzyme helicase unwinds the DNA double helix, exposing the template strand. RNA polymerase then binds to the template strand and starts synthesizing a complementary mRNA strand by adding RNA nucleotides. Each nucleotide in RNA consists of a ribose sugar, a phosphate group, and one of 4 nitrogenous bases: adenine, uracil, cytosine, or guanine. As RNA polymerase moves along the template strand, it reads each DNA base and pairs it with a complementary RNA nucleotide. Adenine in DNA pairs with uracil in RNA, thymine with adenine, cytosine with guanine, and guanine with cytosine. This continues until RNA polymerase reaches a termination sequence, signaling the end of the gene. The mRNA strand is now ready for further processing in eukaryotic cells or direct translation in prokaryotes. In this model, we see RNA polymerase guiding each RNA nucleotide to form the mRNA strand step by step, mirroring the process of transcription. Ribosomes are made up of ribosomal RNA, or rRNA, and proteins that are organized into 2 subunits, the large and the small subunits. rRNA forms the core of each subunit, providing structure and a platform for protein synthesis. Translation is the process where mRNA is decoded to build a polypeptide, or protein, and it takes place in the ribosome in both prokaryotic and eukaryotic cells. Translation has 3 main stages: initiation, elongation, and termination. In initiation, the small ribosomal subunit binds to the mRNA at the start codon, AUG. The first tRNA, carrying the amino acid methionine, MET, pairs with this start codon. Then the large ribosomal subunit attaches, forming a complete ribosome with 3 binding sites: A, amino acyl, P, peptidyl, and E, exit sites. During elongation, the ribosome reads each mRNA codon, and a tRNA carrying the corresponding amino acid enters the A site. For example, here we see tRNA matching codons with amino acids like histidine, serine, and tyrosine. The ribosome catalyzes the formation of peptide bonds between amino acids, creating a growing polypeptide chain that moves from the P site to the E site as each tRNA exits. Finally, in termination, when the ribosome reaches a stop codon, in our model it is UAG, no tRNA can bind. Instead, release factors help detach the polypeptide, ending translation. The resulting polypeptide will fold into a functional protein, completing the process of gene expression.